Let's start with transcription in the nucleus. First, we have to look at our DNA strand. One side of the DNA is called the promoter region, or Tata box, and is where the transcription factor binds to. The RNA polymerase then binds to the transcription factor. It moves down the DNA, breaking the hydrogen bonds between the bases, creating a template strand and a coding strand. The template strand is identified as being the one that goes in a 3 to 5' direction. As the RNA polymerase moves along the DNA, it builds single-stranded RNA by adding bases that are complementary to the template strand, using uracil instead of thymine. The part of DNA that is transcribed into RNA is called the transcription unit. Eventually, the RNA polymerase reaches the termination sequence on the DNA strand, which signals it to stop transcribing and fall off the DNA. Modifications must then be made to the separated pre-mRNA. A 5' cap of 7 guanines is added, and on the 3' end, a poly A tail is created. Lastly, coding regions called exons and non-coding regions called introns must be separated from each other using spliceosomes. The spliced exons, which now make up mRNA, leave the nucleus to start translation. Once in the cytoplasm, a ribosome, which contains A, P, and E sites, clamps to the 5' end of the mRNA. The mRNA has codons, each made up of 3 nucleotides that together code for a certain amino acid. The tRNA has anticodons, which are complementary base pairs for codons. Some have an amino acid attached to them and are called aminoacyl tRNA. To begin translation, the start codon, which is always AUG, codes for amino acid methionine and is recognized by the ribosome. The tRNA that is attached to methionine enters the P site and binds to the codon AUG. The next aminoacyl tRNA enters the A site, and the amino acid forms a peptide bond with methionine. Each time an amino acid is added, the ribosome moves over one codon. When the tRNA is shifted to the E site, it exits the ribosome. Finally, a stop codon is reached. They bind with anticodons of tRNA that don't carry amino acids, which means there is no new amino acid for the chain to bond to. The ribosome subunits fall off the mRNA and the polypeptide side chains begin interacting, causing it to create a 3D structure, forming a protein.